Assalamualaikum and hi, this is a project proposal presentation for STF3013 Final Year Project 1. I will present to you my project that will work on the title Comparative Leaf Proteomes of Irradiated Mutants and Wild Type of Oryza sativa indica. First of all, let me introduce myself. My name is Muhammad KHAIRUL AKMA bin Darwis, a third-year student from Resource Biotechnology program. For my project, I am working under supervision of Professor Dr. Muhammad Hasnain bin Muhammad Hussain. Oryza sativa or rice is a type of grass that produces grains and known for its close relation to Oryza rufipogon, which is a wild grass in Asia. Indica is one of the subspecies of Oryza sativa, grouped under Poales order and classified under Phocaea family. Typically, the cultivation of indica rice mainly can be found in the tropical and also subtropical regions. Indica rice can also be well grown in highland, lowland, and flooded areas, since it can adapt to various types of soils. In most parts of Asia, especially Southeast Asia, indica rice is recognized as socio-economically important and has become a staple food in many countries around the globe. Indica rice plants have an outstanding performance in producing demandable rice grains, which shatter easily, flakier when cooked, as well have higher amylose content. Due to these uniquenesses, it is understandable that indica rice is the most popular rice variety in the world so far. Statistically, indica rice cultivation actually accounts for more than 80% of the world rice cultivation and left other rice varieties far behind. In this project, we will adapt proteomics as the main approach. For the past several decades, Proteomics has been recognized as the most prominent methodology which offers a broad range of applications from conventional to high-throughput techniques. Proteomics facilitates in understanding protein expression, structures, functions, interactions, and also modifications. It is an undeniable fact that all plants in the wild environment are exposed to various types of radiation. The exposure of plants to radiation may result in the occurrence of mutation. Hypothetically, when the mutation occurs, the biological process of plants somehow will be altered. This situation then raises query among public members on the effect of the mutated rice species for the agriculture industry. Thus, this situation arises a question mark that leads to the construction of research question. Does mutation bring advantage or defection to the agriculture industry? So basically, this project has two clear objectives. The first objective of this project is to compare the leaf proteomes between irradiated mutant and wild types of Oryza sativa indica. Next, the second objective of this project is to evaluate the alteration of protein productions in irradiated mutant type as compared to the wild type of Oryza sativa indica. For over centuries, human civilizations has witnessed the significant role of rice as the most prominent food source. It is believed that Asian civilization was developed based on the rice cultivation that spawned a rapid development of empires and dynasties. The domestication of rice has an intricate history and remains debatable from time to time. According to archaeological evidence, China and India are the two nations which are strongly appointed as the centers of rice domestication. As mentioned before, rice plants are grouped in Poales order and classified under the Phocaea family. Rice is a type of annual plant which completes its life cycle within one growing season. Generally, rice plants consist of three basic systems which are root, shoot, and flower system. Rice grain consists of hull, bran, endosperm, and germ. The grain took about 35 days to ripen and mature after the pollination event. Despite the impressive facts which cover 80% of the world's rice cultivation, somehow there is still a lack of research efforts conducted on indica subspecies, especially in comparative proteomics that is associated with mutant type. Conspicuously, protein is the best of the proteomics study. For the past several decades, proteomics has facilitated researchers in understanding various aspects of scientific mechanisms in living organisms. For instance, salt tolerance in okra plant and drought tolerance in maize plant. In rice plants, most of the comparative proteomics study actually focus on the rice adaptation, such as drought and salinity tolerance. In the plant kingdom, the occurrence of mutation unnecessarily indicates a negative phenomenon. Some of mutated rice species actually gave advantages such as high starch content, better yielding potential, and also better energy metabolism. Typically, methodologies that are highlighted in proteomic study are protein extraction and protein suppression. There are many protein extraction methods such as TCA acetone, phenyl extraction, and polyethylene glycol fractionation method. The suppression technique by using two-dimensional polyacrylamide gel electrophoresis provides an efficient and reliable technique based on molecular weight and isoelectric point. The combination of using PEG fractionation and 2D patch gave better resolution without any streaking, thus will be used in this project. For materials, the mutant type of indica rice seeds will be provided by Nuclear Malaysia, Ministry of Science, Technology and Environment, and wild type of indica rice seeds will be provided by Proteomics Laboratory of University Malaysia Sarawak UNIMAS. The seeds will be grown in the growth chambers for about 4 weeks. After that, the indica rice leaves will be obtained from both growth chambers for next process. Here we outline several methods for this project. Firstly, the germination of indica rice seeds will take a duration of 5 weeks before leaf is obtained from the growth chambers. Each of the leaf types will be subjected into the preparation procedure to convert them into a simple form of samples. This can be done by using gentle and complete pulverization on the leaves by using a mortar. The samples then will be subjected to polyethylene glycol fractionation technique for protein extraction purpose. This technique is considered as a very effective technique which offers a very efficient ways of protein retrieval. Next, the extracted protein will be loaded onto the two-dimensional polyacrylamide gel electrophoresis for protein suppression purpose. Here, the protein can be separated according to their molecular size and also isoelectric point. Lastly, the generated results from the protein suppression will produce protein spots. These protein spots will be analyzed by using software. So what we expected from conducting this type of project? From this project, we expected to be able to achieve all the objectives stated earlier on. To add, we also expected to be able to determine the effect of irradiation on the plant rice growth. Thus, from that, we are able to evaluate the usefulness of mutant rice type for the agriculture industry. In addition, this is the work schedule that is outlined based on the main activities that will be done along this project. Finally, I would like to express my deepest gratitude to my supervisor, Professor Dr. Muhammad Hasnain bin Muhammad Hussain, for his precious guidance in supporting me to complete this significant final year project proposal. Hereby, my final year project on comparative leaf proteomes of irradiated mutant and wild types of Oryza sativa indica can be proceeded.